just give it a couple more minutes as people roll in, and then we'll get started.
Mike, I think you're both the first speaker and the moderator, so it's probably good to get started now.
Okay.
Do I have that right, the timing?
Yeah.
Yeah, it's 2. Yeah.
Okay, sweet. Yeah, so like Rich mentioned, I am both the first speaker and the moderator for this session. And for those of you that don't know me, I'm Mike Blazanin. I'm a PhD student at Yale University, and I work with Paul Turner, who's also on the call. So I'm looking forward to telling you all about some of the work that we actually recently published in the ISME journal as a review paper titled Community Context Matters for Bacteria-Phage Ecology and Evolution. So for those perhaps not from the microbiology side, bacteriophages, or phages for short, are viruses that infect bacteria. And so out in the natural world, you might have a phage and a bacteria, and when they come into contact, the phage will attach to the outside of the bacterial cell, then they'll inject their genetic material and hijack all the cellular machinery in order to produce a whole bunch of new phages. And for lytic phages, which is a lot of them, this process kills the host. And so this sort of host-parasite relationship has made bacteria and phages really wonderful models for studying ecological and evolutionary processes. And in nature, bacteria and phages often live in really complex multi-species communities. But despite that kind of reality in the natural world, studies on bacteriophage ecology and evolution haven't exactly matched that kind of natural structure. So largely there are kind of 2 bodies of work that have studied bacteriophage ecology and evolution. The first is the sort of very simple reductionist sort of approaches of running experimental evolution, often in simple laboratory communities with usually 1 phage, 1 bacteria, and flasks or test tubes or something like that. And these sorts of experiments provide a lot of experimental tractability, but obviously don't mirror the natural world very closely. And on the other end of sort of the community complexity spectrum, in the last decade or 2, especially as culture-free sequencing-based approaches have become quite feasible, observations of natural communities, whether it be animal microbiomes or ocean or soil communities, have become increasingly common. But obviously, in those situations, it's really hard to do experiments. And so to kind of fill in the gap between these 2 different bodies of work, recently there have been some studies published doing experiments, but in these medium complexity communities with multiple species to try and bridge that gap. And so what Paul and I set out to do was to review and synthesize kind of that body of research to highlight what has been found so far and what is left to discover, where is more work needed. So we specifically focused on papers that manipulated this community context or community complexity within a single set of experiments. And across kind of both of those levels of complexity, then observed ecology and/or evolution. And so this provides a really powerful way for us to understand what might we have been missing by having sort of really reduced experiments relative to the natural world, and what might be kind of facilitate that translation of results from laboratory experiments out into more natural settings. So the papers that we reviewed and pulled together, we generally broke down the findings into the ecological effects of community context and then also the evolutionary effects of community context. And so I'll walk through some of the main kind of patterns that we found with examples and then also highlight some of the remaining unknowns. So in terms of the ecological effects of community context, perhaps the first and most obvious thing to look at is how does a community context affect that focal bacterial density, right? The phage host, how does that affect the population density of that bacteria? So in this experiment published by Mumford and Freeman in 2017, they looked at Pseudomonas aeruginosa, which was grown alone or with one or another bacteria or all three together. And plotted on the y-axis here is just their density, and I'm just showing the data from their phage-absent treatments. And what we can clearly see from this data is that the community context generally suppresses the focal host density. And this is, as we would expect from resource competition, and was largely consistent across all of the studies that we reviewed that had this sort of data included. Now, what about the flip side, the effect on the focal phage? Well, there the story is largely the same. So this is showing data published by Alseth Percy et al., published in 2019. And here they're showing phage titer over time in two different treatments. One, this black line where Pseudomonas aeruginosa, again, is in monoculture, and in this pinkish line, it's in co-culture with three other bacterial species. And it very clearly shows that phage density is suppressed when you have these other bacteria around, as you would expect from sort of bottom-up control limiting phage growth when hosts are limited. And this, again, was largely consistent across all of the papers that we reviewed. So now, how about interactions? Ecological interactions between community context and phage presence, both kind of simultaneously impacting the phage host. Well, it turns out that in this, findings have actually really varied across studies and across systems. So some of the findings have included things like synergistic suppression of community context and phage presence, where each individually just suppresses the focal bacteria some, but together they actually drive the focal bacteria extinct. Other findings have included that these effects can work largely independently, where both maybe suppress the focal bacterial density, but they don't really synergize in any way. And even in some cases, the community weakens or reverses the effect of phage, where phage will reduce the bacterial density, but then in the community context, it actually has a much weaker effect. And so clearly we need a lot further work to start to resolve how and when and what factors underlie this interaction between community context and phage presence. Now going on to the evolutionary effects of community context, a lot of papers naturally will look at the evolution of bacterial resistance to phage infection. And so we also kind of summarize the effect of community context on the evolution of that resistance. So again, showing some data from Mumford and Freeman, published in 2017, they, among other data that they showed, they measured the percent of populations resistant to phage after evolution in one of four treatments. So monoculture, co-culture with Staph aureus, co-culture with Staph multifilia, and co-culture with all three. And this is just showing the co-phages data, and you can very clearly see that evolution treatments where other bacteria were around, the evolution of resistance was constrained. And this kind of constraining effect of community context was something that actually emerged from several different papers, where the evolution of resistance is retarded or slowed by the presence of other bacterial species. Now, what about the mechanisms by which resistance evolves, right? Bacteria have a multitude of mechanisms through which they can evolve resistance to phage infection. And one paper has actually looked at this. So Alseth Percy et al. Published in 2019, this great paper comparing CRISPR-based resistance to resistance via mutations in surface proteins, the surface proteins that phages use to attach. And here they've plotted the CRISPR relative fitness compared to those surface-based mutations on the y-axis. and across the different evolutionary treatments, where again, monoculture, co-culture of Pseudomonas ruginosa with these 3 different bacteria, and then a mixed community. And what they very clearly found was that community context favored CRISPR-based resistance over this surface mutation-based resistance. And what's still unclear is what the effect of community context is on other resistance mechanisms, like I mentioned, because there are many, many of them, and nobody has really tested it. So next, kind of flipping to the other side of the evolutionary battle between phages and bacteria is, how does community context affect the host range evolution of phages, in particular their ability to evolve to infect bacteria that were previously resistant? So Desordi, Kana et al. Published in 2017 this study where they took 1 phage, 1 susceptible bacteria, and 1 initially resistant bacteria, and then they inoculated them into flasks, sterile mice, or mice with an intact microbiome. And then they checked for when will the phage evolve to infect this initially resistant bacterial strain. And what they found was that the phage host range only expanded in the mice with the intact microbiome, right? So in their case, they found that community context actually facilitated this host range expansion by the phage during experimental evolution. But again, we don't know how general this finding is. They are the first, and as far as I'm aware, only people to document this sort of study in a community with 3 or more species. And so we need further work to kind of delineate what the boundaries and conditions are when community context will facilitate host range expansion. So finally, how does community context interact with traits that aren't just infectivity and resistance? Right? These sort of pleiotropic consequences of adaptation, whether it be growth rate or yield or other traits. And again, this is an area where studies have really diverged. So findings in sort of this trade-off, trade-up space have included things like community context enhancing the costs of evolving resistance, cost being here measured as competitive fitness, growth, things like non-interacting costs, where Mumford and Freeman observed that adaptation to a community carried costs, adaptation to phages carried costs, but these costs tended to not interact. They just sort of combined additively. Others have found no pleiotropic effects, at least in the phenotypes that they measured of community context. And even in some cases, community context driving pleiotropic benefits, like favoring the evolution of higher fitness when compared to evolving in the absence of community, or facilitating evolutionary movement along trade-off fronts, kind of facilitating movement along constraints. So to kind of step back and wrap up, there are clearly some convergent findings emerging, like the suppressive effects of community context on ecology and on the evolution of resistance, but there are a lot of open questions remaining. So some of these questions include things like what drives those ecological interactions between when both community members and phage are present? When does community context alter coevolutionary dynamics, things like arms races? How does community context affect mechanisms of resistance other than CRISPR and surface-based mutations? Under what conditions does community context favor host range expansion by phages? What sort of factors might determine the pleiotropic costs or benefits of adaptation to a community context? And lastly, but perhaps not least importantly, how do these findings hold up as we kind of expand this space that these experiments take up? A lot of these experiments have been done over just 15-ish generations of evolutionary time and in communities with like 4 or 5 species. And so it'll be really interesting to see going forward as more people do experiments in more speciose communities and over long periods of time, how well these patterns kind of persist. So again, I'll point you to the review article that was recently published in the ISME journal, and I'm happy to take any questions. Rich, you have your hand raised. Go ahead.
Thanks. Very nice talk. I was wondering that one of the counterintuitive ones was the one where the community promoted the evolution of the extended host range phage, if I understood it right. That kind of surprises me. But did they look at was it only looking at this target bacterium? I guess it was the green one, so to speak, in your image. Or did they sort of ask whether that was facilitated because it was also infecting many of the other members of the community, so that it just kind of blew up its population size and therefore allowed wider exploration?
Yeah, so I mean, kind of their metric was whether it would infect green, but yeah, they did kind of dig into it and find that the phage was infecting some of these hosts, and actually that adaptation to some of the microbiome hosts kind of acted as a stepping stone, facilitating it to eventually infect the green host.
Cool, that makes sense, thanks.
Yeah. Pull up the chat here. Let's see. Ben has a question in the chat. The Desordi et al. Result was very interesting. Is there evidence that host range shifts in the phage occurred to some of the native microbiome? Yes, so exactly the same question. Yes, the native microbiome served as a really nice stepping stone, as they showed. And I'd encourage you to check out that paper because it's really cool. Anya has a question in the chat. Did you look at all if questions have been examined with modeling? We looked very briefly, but honestly, the modeling space on this question is enormous, right? If you start thinking about anybody who's done experiments with Lotka Volterra and asking sort of competition predator or competition parasite relationships, it just got way too big for us to feasibly review in this paper as well. I would have loved to, but yeah, it just kind of, we scratched the surface and we're like, man, that's a whole separate review paper on its own. I think there's time for maybe one more question. Otherwise, I will stop sharing and let Sylvie start getting set up. Great. Sylvie, you can go ahead and share your screen because you are next.
Hi. Can you all see my intro slide?
Yep, looks good.
Okay, great. Well, it's great to be back at Beacon. I really appreciate the opportunity. And so today what I'd like to tell you about is some work I've been doing looking at the rules of microbial community assembly. So microbes and more broadly microbial communities play important roles in the environment, in industry and health. And their roles can be positive, such as providing essential nutrients to their hosts, or negative, for instance, by making their hosts sick. And so given their importance, one of the main aspirations of microbiome biology is to be able to predict how microbial communities assemble, how they respond to perturbations, and ultimately how they evolve. And answering these questions is important to be able to efficiently manipulate and design microbial communities that perform desired functions. But one can only aspire to predict microbial community assembly if it is reproducible. So for instance, if the patterns of community assembly we see under certain conditions are not repeatable, but instead we always get different patterns, then there is little we can predict. And so then the question becomes, so how reproducible is microbial community assembly? And also what is reproducible, what isn't, and why? And so to address this question, we use an enrichment community approach, which basically consists in watching multiple replicate microbial communities self-assemble in well-defined and controlled habitats. So in a nutshell, we first collect a sample from the natural environment, say the soil, then we bring it to the lab, and then we inoculate a sample which contains a highly diverse pool of species into well-defined synthetic media in multiple replicates. And then we let them grow, and then we come back and dilute a small fraction into fresh media, and then we repeat this growth dilution cycle multiple times. And then at the end, so once the communities have reached equilibrium, we assess the taxonomic composition using 16S sequencing. And then all this is done under well-controlled lab conditions. So then the first question we asked was, so how reproducible is microbial community assembly in these synthetic identical habitats? And so what I'll show you now is the result of an experiment that was started with 12 different species pools collected in different locations, so in New Haven and West Haven, and then each was inoculated into fresh minimal media with glucose as the single current source and in 8 replicates each. And then we passage these to fresh media every 48 hours for 12 transfers. And so shown here is the species level taxonomy composition after 12 transfers. So for the 12 inocular times 8 replicates, so that's 96 communities in total. And then what we can clearly see is that there is a high variability even for samples that were inoculated from the same pool of species. But when we looked at the family level composition, we find that there is a strong convergence regardless of the initial inoculum, and with most communities being dominated by 2 families, so the Enterobacteriaceae and Pseudomonaceae family. So these observations really led to 2 fundamental questions. That is, so why is community assembly so convergent at the family level? And also, why is community assembly so variable at the species level? So for the sake of time in this talk, I'll only focus on the first question. So why is community assembly so convergent at the family level? So our hypothesis was that this family level convergence reflects the emergence of functional convergence, which would then suggest that the metabolic traits that are being selected are conserved at the family level. And so to test this idea, we isolated close to 80 strains from the self-assembled equilibrium communities. And these span across multiple families and genera, which are highlighted here in this tree. And so here I'm only going to focus on the 2 dominant families, so the Enterobacteriaceae and Pseudomonas. So what we did first was to grow these strains separately in M9 plus glucose for 48 hours and then measure their growth rate. And the question here is, how do these 2 dominant families grow on glucose? And so in this plot here, each point corresponds to a different isolate. And so what we find is that the Enterobacteriaceae grow on average faster than the Pseudomonas, which may explain why they are more abundant in the communities than the Pseudomonas. And it is known that selection for fast growth on glucose leads to the excretion of metabolic byproducts like acetate or lactate. And so what we did next was to look at the supernatant of these isolates. And what we find is that after 16 hours of growth on glucose, which is approximately when most glucose has been depleted, all Enterobacteriaceae secrete significant amounts of acetate, lactate, and also some succinate, while the Pseudomonas do not, and even later in the growth cycle. And so here it's important to emphasize that all the Enterobacteriaceae belong to, they belong to different genera, which leads to some differences in the quantitative details, but overall they all have very similar secretion patterns, and especially for acetate, which is the dominant byproduct. And so what this suggests is that this type of niche construction, in a sense, is conserved quantitatively at the family level. So far we know that the Enterobacteriaceae grow faster on glucose compared to the Pseudomonas, and they do so while secreting lots of organic acids. So then the natural follow-up question was, so do the Pseudomonas in turn grow faster on the secreted byproducts, which could explain why they coexist with the Enterobacteriaceae? And the answer is yes. So the Pseudomonas grow on average faster on the 3 dominant byproducts. And so together, what these results are showing us is that they strongly support our initial hypothesis that family convergence reflects a functional convergence. And so here is what we think is happening. So first, there is strong selection for fast growth on the supplied glucose, which is conserved at the family level. Then this leads to the secretion of convergent levels of organic acids in the environment by the Enterobacteriaceae. And then the Pseudomonas will prefer organic acids to glucose and are also better at growing on them. They cross-feed on these organic acids. And so what this means is that this family level convergence in glucose habitats reflects an emergent self-organization between two functional groups. The glucose specialists will ferment the glucose, and then the organic acid specialists that mostly feed on and respire the byproducts of fermentation. And so now I'll call the glucose specialist fermenters and then the organic acid specialist respirators. So F and R. So we know that what microbes do when they are alone, so as single species, is not necessarily what they do when they are together with other species. So then the next question was, so do these metabolic rules apply in the context of the communities? And so to test this, we grew the communities in glucose and then tracked the ratio between respirators and fermenters, as well as the concentrations of glucose and acetate at different time points during a 48-hour growth cycle. And so what I'm showing here is the result of one representative community. And so consistent with our hypothesis, we find that fermenters have a growth advantage early on the incubation, so as the glucose is being depleted, and this can be seen by decreasing the RF ratio, while respirators have a growth advantage later on when the glucose is absent, but the acetate is abundant, which we can see by an increase in the RF ratio as the acetate is being depleted. And so this just provides further evidence that this family convergence reflects an emergent metabolic structure between these two functional groups. So the next question we were interested in was, so can we quantitatively predict and explain the ratio between these two functional groups? And so the communities converge to a RF ratio of 0.29, but why this ratio and not something else? And so our first thought was, so can a simple resource partitioning model actually predict this ratio? And so for this, we developed a simple model, which basically just assumes that all supplied glucose goes to the fermenters, and then that all secreted acetate goes to the respirators. And so then the RF ratio is basically the ratio between the average biomass that respirators can extract from the secreted acetate and the average biomass that the fermenters can extract from the supplied glucose. And so then the predicted ratio is given by this equation here, so defined by these three parameters. And one of the advantages of this simple model is that we can actually measure these parameters experimentally using our collection of isolates. And so then we did so, and then we input those in this equation for all respirators, fermenter pairs. And what we find is a predicted RF ratio of 0.31, which is very close to the experimentally observed ratio of 0.29. We also estimated this ratio using flux balance analysis by estimating these parameters using a library of published genome scale models for close to 70 Pseudomonas and close to 60 Enterobacteriaceae strains. And we again find a very similar RF ratio. And so just to summarize what I've shown you today is that family convergence in glucose habitats reflects a convergent metabolic self-organization between two functional groups, so glucose specialists and organic acid specialists, and also that simple metabolic models can be combined with enrichment community experiments to predict and explain the metabolic structure of communities. And so I just really want to finish by showing you some new results. So this is from a community assembly experiment that we have done for close to a year. So we started with 12 replicate communities, and then what I'm showing you here on the left is the community composition for 6 of those replicates across time. And then on the right is the RF ratio for the 12 replicate communities. And what we can see is that the RF ratio remains more or less constant over time. And we are now looking at more into detail of what's happening through time by looking at how growth parameters and metabolic secretions are changing through time. Yes, so I'd just like to finish by thanking people who were involved in all this work and funding. And yeah, happy to take any questions.
Great. If you have questions, they're welcome to unmute, or you can just put them in the chat and I'll read them for Sylvie. Yeah, Emily, go ahead.
Fantastic talk. This is so cool. It like gives me hope that like our modeling of this stuff can actually predict what will happen. So in the long-term experiments that you talked about at the end, you said you're seeing change over time in what they're releasing. Is that because of additional kind of sorting within which species are in the community, or are they actually starting to evolve? Or do you know yet?
So all the communities were started with the same minotaur. But it looks like, so we started looking at into the isolates, and it looks like they evolved. So there is some changes. So we have some data showing that there is a change in the lag phase of some of the isolates, and so they are fermenters are evolving a shorter lag phase, and we are also seeing changes in the secretion profile. So it's evolution is happening, but we are still looking into this. It's still very new.
Awesome. I'll be excited to see what happens.
Great. Well, as always, great talk, Sylvie. And let's switch over to Shakeal. And any further questions, feel free to put them in the chat, and I'm sure Sylvie can answer them there. Okay, great. Take it away.
So, hi everyone. I'm Junyuan, and I'm his Shakeal. And we are going to present our project this summer: more symbionts, more problems. Evolutionary stability of host endosymbiont mutualism is decreased by multi-infection, led by Emily Dawson and Aya Worcester. So first, we're going to provide some background information. We all know mutualism is very important to humankind. Pollinators and soil microbes are critical in agriculture. Bacterial microbes regulate our health, and domestic animals provide us with food. So we want to understand how mutualism is involved in the nature. We think because the humongous microbiome actually mutate in a relative quick rate, we do believe that this is a good choice to understand how these things are involved. So we believe that mutualism is involved from a spectrum from parasitism to mutualism. Mutualism in our experiments is defined as host and symbiont cooperate with each other and both benefit. Parasitism, endosymbiont steals resources from the host, and the host expends some resource attempting to defending themselves. The model or the platform we use is the simulation, which is a software model of symbiosis and allows the evolution of parasitism and mutualism. In the model, each host has several things within it, and the behavior of the host or symbiont is defined by interaction wall. When the interaction wall is greater than 0, the host or symbiont is mutualism. And when the value goes to below 0, they become parasitic. In the simulation, it contains a word of the host and endosymbionts pair we mentioned in the last slide. And it has several updates. In each update, the word will provide some resource to the symbiont, and the symbiont will try to, if mutualism, try to get some resource from the host, if parasitic, try to steal some resource from the host. And when the host has certain amount of energy, they try to reproduce. When they reproduce, all the settings are the same. The offspring had all the setting of the offspring is the same to the parent. Expect the interaction wall can mutate. And when the host is trying to reproduce, the symbionts also get a chance to go to the offspring of the host. We call it vertical transmission. And the chance of whether the vertical transmission success is called vertical transmission rate. So the previous research from Aya Weiser has shown that the vertical transmission rate we just mentioned is really important to whether the mutualism can evolve. So, but in that paper, it only has the case when each host can only have one symbiont in it. So we're really interested in what happens when multiple symbionts can infect a single host.
So what happens when multiple symbionts infect a single host? We're going to just look at Anja Wassenaar's results very briefly. So as we can see here, we have the final mean interaction value on the y-axis and vertical transmission rate on the x, and the host is in red and the symbionts are in pink. And this was run for 100,000 updates, and we can see that there's a relatively smooth, clean transmission of as the vertical transmission rate increases, the final interaction value also increases. So we were interested to see if this relationship would persist as we added symbionts to the host. So in our first run, we have 10 symbionts per host, and we're still showing from 0 to 100% vertical transmission rate. So we can see from about 0 to 0.4, the vertical transmission rate, the hosts and the symbionts are parasitic. And we were interested in why this was happening. So we zoomed in on one of the results. And we have two charts here with the symbionts and the host. And we have the parasitic and detrimental colors are the reds and the purples and pinks, and the mutualistic colors are the greens and blues. So we can see for the symbionts, they're mostly blue or mutualistic with some striations of parasitism, which is what we expected. And for the host, it's mainly mutualistic still. So we wanted to see if this would change if we added more symbionts. So next we have 20 symbionts per host, and we can see that there's a shift beginning to start where the final mean interaction value, it's taking more, it's taking a higher vertical transmission rate for the interaction value to kind of pass that threshold. And as we add 40 symbionts, you see that that threshold increases and to 50. And the same thing continues at 60. And it seems like there's just an increase vertical transmission rate necessary, and all the way to 100 symbionts per host. We see that even when the vertical transmission of the symbionts are guaranteed, there's still a parasitic relationship between the hosts and the symbionts. So we wanted to see what was happening among the replicates here. So we zoomed in, and we found that this is very different from with only 10 symbionts. So we see that most of the symbionts here are parasitic with a very thin band of mutualism on the bottom. Excuse my siblings, I'm sorry. But most of the symbionts are parasitic, and this is also true for the hosts. So we could see that as the number of symbionts per host increases, there's a breakdown of cooperation amongst the symbionts within the host, which leads to parasitic interaction value. And this relationship continues for the host as well. So some of our main takeaways here: increasing the number of symbionts per host decreases the cooperation between the host and symbionts. And this leads to a decrease in mutualism between the host and the symbionts, even when the vertical transmission rate is guaranteed. And we think this is because there's a small number of parasitic symbionts, but the host can't treat the symbionts any differently. So this kind of messes things up for all of the symbionts and leads to an overall increase in parasitism. So we were interested in seeing if we altered the interaction between the symbionts, if this would change anything, and Jen's results will go into that.
Right. So in order to give the symbionts initial incentive for cooperation, we add a public good game. The public good game means that, for example, in a society, everyone should donate some amount of their money to a public good, for example, a park. And after the public good is accomplished, it will return averagely some points of money back to each person, often with some multiplier. So in our system, this is going to be first, when the symbionts get some resource from the host, they will also donate a certain amount of their resources to a resource pool. And after each sim in the same host has donated their resources, the pool will averagely return each sim amount of energy with some multiplier. And we call the initial donation, the initial part of energy donated to the pool donation rate. And when we do the experiments, we do the same 10 symbionts per host and 100 symbionts per host. In the 10 symbionts per host, we can see the host is always mutualism, and this is no difference to the previous experiments where the Shakeal has shown. But for the experiment for 100 symbionts per host, everything changed. Compared to the previous experiments, we found that even at the vertical transmission 0.06, which is this graph is showing, at the initial donation rate 0.8, the host can be mutualism. But in the original experiments, no matter how vertical transmission rate is, the host is still parasitic. So we zoomed in to see what really happened. In the graph, we can see that most of the hosts are mutualism, except there is some gradually changing layer happened in certain replicants, and also it's the same for the symbionts. So the conclusion is, first, for experiments that has 100 symbionts per host, public good games make the host more cooperative. And this is also can be shown in the zoom in version of the graph. And second, in some replicants, the cooperation appears unstable because there is some gradually changing layer happened in the, for example, replicant 610802. It is really hard to say when the evolution will end and how the final interaction value or how the final behavior of the host will be. And it's natural to think this is the influence of donation rates, but actually, when we plot the donation rate of all the replicants, it seems like all the donation rate finally becomes the same, and we cannot say whether the donation rate has certain influence on this gradually changing layer. So this is something we're going to experiment and try to explain in the future. And thanks to Emily Delson and Al Wester and the founding KR Fellowship. Thanks all. Questions.
Awesome, thank you. It looks like we have like 2 minutes roughly, so people are welcome to raise your hand and unmute to ask a question or put it in the chat and I can read it live. Well, if no one else has a question, I actually have a question for Shakeal. When he was showing those kind of some of the very first symbiote kind of patterns, and it looked like there was like a progression, almost like a takeover. Yeah, go back one. Keep going, one more. Yeah, there. Yeah, so in some of those, it looks like over time you're transitioning from cooperative to parasitic, but it doesn't seem like the same thing is happening in the host. So I was wondering if you could just kind of dig in what's going on more in those runs compared to the others where it seems like the neutrality is persisting.
Well, in these runs, when there's only 10 symbionts in the host, that's still a pretty, well, it's kind of manageable for the host. There aren't very many parasitic symbionts in this one, so it wouldn't have as much as an effect on the host, since they wouldn't need to cooperate as much. The parasitic ones are kind of taking over because they're stealing resources from their fellow symbionts, I suppose. That's why we see that kind of takeover there. And it's just not affecting the host as much because the host still has resources that it's giving evenly. So it still has enough for itself. And they're not parasitic enough to affect the host and make it go on the defensive initially. Yeah.
Gotcha. Cool. All right, well, I think in the interest of time, we'll switch over to Eric Libby, and any further questions, again, you can put in the chat. I'm sure Jen and Shakeal would be happy to answer them there. So go ahead, Eric.
Great. Can at least one person see my slides? Okay, good. That
looks good.
Yeah, thanks. So first of all, thank you so much for inviting me and letting me have the opportunity to talk to you about this. It's extremely exciting. But despite that excitement, I accidentally chose the most boring talk title I've ever chosen in my life. So I'm hoping that this talk will make up for what is going to be a rough start. So before I begin, I wanted to thank the collaborative team. This is a project with my PhD student, Rebecca Anderson, Chris Kempes, who's at the Santa Fe Institute, and Jordan Oki, who's at Arizona State University. So this work begins with an observation that many people have made. If you were to look across the domains of life, you would notice that all the examples of really big things seem to be eukaryotes. And this has led to sort of a common argument, which is that if you want really big sort of complex organisms, you need to be eukaryotes. And they link the eukaryogenesis, the origins of eukaryotes, with the origins of complexity. The arguments usually go as follows. All complex things are big. If you want to be big, you need to have lots of energy. And hey, isn't it funny, but eukaryotes get their energy from a special organelle called mitochondria that's not present in other domains. Of course, we all know that there's some more interesting things going on here, that the mitochondria used to be organisms, and an endosymbiosis is what made them organelles, and it's believed to be a very rare event. Now, this last bit it's hard to sort of estimate how rare it is because obviously a lot of lineages are lost, but there have been some people that think it's only a singular event, or at least just a handful of times. Certainly, if you were to look at endosymbiosis in general, it seems to happen far more frequently in eukaryotes than in prokaryotes. So there's many possible reasons for the rareness. So I would try to move my window. So yeah, many possible reasons for the rareness. The limited space, like inside cells. There's ecological opportunity, where maybe you need two cells of a very specific type. Evolutionary maintenance, it might just be hard to maintain the relationship over several generations. Physical barriers, there might be something about the cell structure or cytoskeleton that makes it hard, or maybe cells haven't evolved predation or the ability to engulf other cells. And environmental conditions, maybe you need a very specific environment, some mood lighting or something that would encourage an endosymbiosis. The problem though with these hypotheses is that they're all verbal arguments, they're very qualitative, and there's no real way to compare them or to quantify them or to even include them in some framework. And so yeah, this calls for a null model, and that's really the aim of this collaborative project, is to come up with some theoretical framework that gives us some expectations of why prokaryotic endosymbiosis might be rare and if there's certain factors that influence it. So we were inspired by the Drake equation. So that's with Frank Drake, the astronomer. I always get him confused with Francis Drake, the pirate, but they are very different people. The Drake equation is used to calculate the number of civilizations, alien civilizations, that we might be able to communicate with. And it's basically a product of a bunch of terms. So it's like the average rate of star formation, the fraction of those stars that have planets, the likelihood that those planets might have living things on them, et cetera. But the basic idea is not that you can calculate the number of alien civilizations that might be out there. Its idea is that you can identify the relevant factors, and then you can try to estimate them, and then you can see how they interact or how they might limit the total product. And that's really what we're taking from the Drake equation. So we tried to come up with a Drake equation of our own in some sense, and this is just a first pass. But we have some initial probability that there's a host and a future endosymbiont in the same environment. Then, of course, there's some probability that the endo, and endo here is just short for endosymbiont, that the endo gets inside the host. And of course, there's some probability that the two of them, the system, can somehow propagate or vertically transmit or produce offspring that can survive for some length of time in the environment. And so we're going to focus on the middle one first. And the reason is because there's two reasons, one of which is that it informs the other approaches, but also because many people think at some level that lack of space might be a driving factor in the likelihood of prokaryotic endosymbioses. So here's the issue of space. If you want an endosymbiont and host pairing, ideally the host should be larger than the endosymbiont. Otherwise it wouldn't work. But of course, being larger than your endosymbiont is not enough, right? A human might be larger than a dog, but the dog is not necessarily going to do so well inside the human. And the reason is because the human body is not just empty space, at least in most cases. It's filled with organs, and these organs would be displaced by the dog. Right? And you could do the same sort of thing with a cell. And it's actually a little bit easier to do in the terms of prokaryotes because we can identify what parts of the cells, what volumes might be used for certain key physiological functions. So DNA, there's volumes that are dedicated towards ribosomes, towards proteins, et cetera. And so what our idea was is that maybe we can estimate each of these sort of volumes, and whatever is left over might be the available space for endosymbionts. And we can get some estimate of how likely an endosymbiont would be able to fit in that space, or how many of them would be. So it turns out that one nice thing as well is that measuring these sorts of volumes, these ones that are over here, I don't know if you can see my mouse, is facilitated by a research field called allometric scaling, which shows that certain key volumes of cells actually scale with cell volume. And this sort of makes sense. What this means is that if you have bigger and bigger prokaryotes, they obviously need more and more space to house their ribosomes, house their proteins. But you can actually relate these volumes by some sort of power law. And so it turns out that for each of those little components that we had just shown you, there's some power law that we can use. And what this means is that we can pick particular host cell volumes, so pick a cell size, and then we can calculate how much of its volume is free for an endosymbiont to move in. And we assume that there's also just a tiny bit of leftover space for water. That's this little gray dash line, and it turns out we're just guessing at 20%, which would mean it would have roughly the properties of honey. But if you have any better idea of how to do this, that would be great. It turns out it's a really tricky thing to try to estimate exactly how much water something needs to still be alive and reproducing. So ultimately, we can put this type of information together, and we can generate a plot like this, where it shows the volume of hosts on the bottom, the volume of endosymbionts on the vertical axis. And for each point here, we can just try to fit as many of these endosymbionts into that host as possible. And that's what we depict in these shaded regions. And so obviously, if you have a really big host, you can fit a lot of tiny endosymbionts. What you should notice is that there's an identity line in red, and things can't go really close to the identity line because obviously this is where the issue of physical space is really operating. Okay. So after addressing this middle area, this idea of physical space, we decided to go to the probability that a host and endosymbiont might be in the same environment. Again, we're fortunate that we can use these allometric scaling properties. So there's been many different studies where people have just sampled things like ocean water and counted the number of cells of different sizes that they observe. And what you see here is you see data from this Cavender-Beres paper in blue, and the red is some sort of power law fit to it. And so what we can do then is we can pick cells of different size and just ask what's the probability that these two cells might be in the same sample. And again, we can get one of these plots where each point here is the probability that an endosymbiont of a certain volume and a host of a certain volume would be present in the same environment. What you see is that the most likely place is at the bottom left. The bottom left is where you have small organisms, and it's simply reflecting the fact that small organisms are more often present than larger organisms. And the symmetric aspect here is just because, again, the host and endosymbiont could go either way when we're just asking if they're both present. So this leads us to the last little area, which is whether or not host and endos can vertically transmit, they can propagate in the future. So to do this, what we wanted to do is we wanted to ask a simple question, which is if an endosymbiont jumps into a host and that system reproduces, is that host and endosymbiont pair likely to produce other daughters, or is the endosymbiont lost in some lineages, or does the endosymbiont reproduce so much that it exceeds the limits, right? So that's that first plot that we did for calculating the limits is actually used in this as well. And so we did a simple branching process. We're often inspired by the plasmid literature here. So here you just think of the little circles are host cells, and the numbers represent how many endosymbionts they have. And all we can do is we can track as the endosymbiont reproduces and the host reproduces, and then you have unbiased or random segregation events. We can track the likelihood that you maintain certain endosymbionts and certain numbers in hosts. And here, these lambdas, which are not that important, are just indicating that hosts might have different growth rates. But for lots of the studies that we've done, we looked at endosymbionts having different growth rates. So one thing that came from these analyses was fairly obvious, I think, to many people that study plasmid dynamics, but was sort of a surprise for us, which is that if the endosymbiont grows slower than the host, it's extremely easy to lose it. And even if they grow at the same rate, the loss rate of endosymbionts is just huge. It's only when you have the endosymbiont growing faster than the host that you're able to sort of maintain it in the population. This is, of course, in the absence of fitness considerations. So the trick here, and I just want to reiterate this, is that if the endosymbiont grows more slowly, you're much more likely to lose it. Well, the funny thing about prokaryotes is that as the prokaryotes get larger and larger in size, they actually reproduce faster. So that means that if you had a small cell and a big cell, the big cell is reproducing faster, which means that that small cell that's inside is unlikely to reproduce before the host reproduces. And so you can lose the endosymbiont very easily there. And so if we do these same kind of plots that we've done before, what you see is that the most likely pairing of host and endosymbionts that maintain the relationship actually needs to have host and endosymbionts in as close a size as possible. Because if there's any difference in the size, the endosymbiont can be lost. So what we can do is, right, we have these three plots for these three different probabilities, and now we can multiply them together to see what areas are most likely to result in a prokaryotic endosymbiosis. And this is what our final plot looks like. What's interesting about this is that if you remember from the first slides is that if you were to say that physical space was a limiting factor, you would see a very high likelihood down here because this is where you have the most room. But the problem is that endosymbiont loss rate is so high that actually you end up, and the likelihoods is so much greater for smaller organisms that you end up shifting the most likely combinations down here. And so for us, what this does is this points to some possible reason for the rarity, because now we can go back and look at different ecological or environmental conditions to try to understand when these types of settings might occur. So with that, I'd like to thank my collaborators again. It's a great team, a lot of fun, and of course, funding from the Swedish Research Council. And if anyone's interested in a postdoc, there's a lot of cool projects related here, so just contact me. And thanks.
Awesome, great talk, Eric. I see that Rich has questions, so Rich, go ahead.
Yeah, very nice. This is kind of probably a silly thought, but the idea that the endosymbiont actually has to be smaller. I was just looking at some videos of, and these aren't the relevant organisms, but didinium consuming protozoa. And I think the didinium, when it's not full of the protozoa, is actually a bit smaller than its prey. Because it can expand. And anyhow, I'm wondering sort of predator-prey interactions or various consumptive interactions and their roles possibly in all of this. That's
great. We actually haven't thought of that. I mean, actually, there's directions on predation that we were going, but we've been kind of, I don't know what the word is, we've been very careful to avoid using the term predation because there's some people in the community that will immediately object about whether or not certain prokaryotes have evolved predation, but we haven't thought of the ability of cells to expand dramatically in size. And that's definitely something that we think would be a really cool direction to look at. Thanks, Rich.
There are several questions in the chat. I think we have time for one of them from Anya. Have you thought at all about how a host might be able to make room for an endosymbiont? She's thinking about how a lot of internals are moved around during pregnancy to make room for an endosymbiont then.
No, I have not thought of that, but that is mostly because that seems like a very tricky thing to try to understand. But I think that it's an absolutely great question. I mean, there are other things that we've been thinking about, which is sort of along the same lines, which is what is the variation in the volumes that you need? So during a lifespan, can you shift these such that you might be able to gain an endosymbiont? But no, but that's a really great question. And I'm thinking of all these terrible medical diagrams of what happens to a woman's body during pregnancy, and it just looks uncomfortable. I just ate a big meal too, so this is dinner time for me, and I'm not in a good place. But thank you, that was a great question.
Cool. Well, I'll let you answer the other questions in chat as we transition over to Nicole. And actually, Nicole is having some technical difficulties, so I will actually be running the slideshow, and then she will just be telling me when to get the next slide. So, Nicole, and just say next slide whenever you want.
Okay, thank you very much. Can everybody hear me?
Yep.
Okay. Hi, my name is Tinyiko Maswanganye, and I'm sorry I can't show my face. I'm talking through my phone. Today I will be talking about determining evolutionary mechanisms of SARS-CoV-2 waterway surveillance. Next. Severe acute respiratory syndrome coronavirus is a virus that causes a respiratory disease called coronavirus disease 19, COVID-19, of zoonotic origin, as we all know. Since the start of the pandemic, COVID-19's rapid spread has resulted in a global health disaster and considerably economic loss, which has impacted most of us now. In order to understand the rapid spread, we have to understand how the virus spreads. To start, next, I will describe the virus structure. The virus encodes 4 structured proteins, which is the S protein, spike, membrane, nucleocapsid, and envelope. The ACE receptor, as shown in the image, was identified as the entry receptor for SARS-CoV-2, and the S glycoprotein binds to the ACE2 receptor with high affinity. The virus enters the host cell respiratory epithelial cells using the ACE2 receptor, transmembrane protease, and serine 2. Here, it suppresses the induction of the antiviral type 1 interferon alpha and beta. Using the viral transcription, the open reading frames are translated into protein in the cytoplasm. If it is successful, the structural proteins will be localized in the Golgi intracellular membrane and will launch the new variants that allow the assembly of the complete genome RNA release from the cell. The ACE2 receptor is highly expressed in the gastrointestinal tract, which may explain the existence of the viral in stool. It is also expressed in the kidney, but there has not been any evidence of CoV-2 in urine. Next. So for this study, we'll be using waterways-based epidemiology, WBE. This study is an effective monitoring method that can help in community-based screening and prevention of emerging and re-emerging diseases. Therefore, using the water-based epidemiology, it is essential in helping us track the spread of the virus across the world and serve as a preventative measure to tackle this global pandemic. Waterway surveillance for SARS-CoV-2 is essential at high-risk settings such as schools, nursing homes, and prison. This will implement large-scale clinical testing in those areas to prevent further spreading. This method is noble at disseminating accurate health statistics to the general public, and the application is adequate in recording the prevalence of SARS-CoV-2 concentration in waterways and the link in the frequency of virus shedders in the population. Using the research strategy, we'll continue to broaden the scope of surveying the population testing to reduce unwarranted clinical testing. Next. The research objectives for this study are to evaluate the prevalence of SARS-CoV-2 in waterways from selected dormitories across the campus of North Carolina Agricultural and Technical State University, identifying SARS-CoV-2 variants and monitor how they evolve, which is still in progress. Evaluate SARS-CoV-2 RNA in present waterways from dormitories across the campus of North Carolina Agricultural and Technical State University, which will enable us to identify symptomatic and asymptomatic COVID cases. COVID-19 cases. Next. To begin the study, we will focus on this map. This map is showing the campus of North Carolina A&T State University, which will be our area of interest. The surveillance of SARS-CoV-2 was conducted in 12 identified high-risk dormitories across the campus of North Carolina State University. 12 dormitories were marked as high risk due to elevated COVID-19 cases during the fall semester, which is in 2020. These dormitories serve a large population of the campus community, and surveillance of these 12 dormitories will mitigate the implementation of COVID-19. Next. This image here, you can also press the other one. This image here, the one with the gray box, demonstrates the setup around our manholes that we have. At each potable hole, looking at the image with the white veil, it's how each manhole is set up. We have a potable pump that pumps the water out of the manhole, which is then transferred into a transfer bottle. And the gray box demonstrates the current setup we have at 3 of our top risk high sites. The sites were set up with the Isoco GSL compact autosampler to collect samples over time. The collection of the samples over time allows us to predict levels of SARS-CoV-2, which will enable quality modeling. Next. The presence of inhibitors in the environment, environmental materials generally makes RNA extraction more difficult to predict. So to protect the quality of viral RNA for a real-time qPCR analysis, the purity and concentration and integrity and contamination risk of extracted RNA, we followed this procedure. After collection of the samples, the waterways was transferred into 50 ml conical tubes where they will be split in two, resulting in a collection of 1 ml of nucleic acid to follow, as shown in the image. Next. The same steps were described before are then repeated in a smaller scale. A total of 40 microliters of total nucleic acid will be collected for real-time PCR. This method has shown to achieve consistent recovery rates and significant reduction in PCR inhibitors. Next. The gold standard for pathogen identification and quantification is still considered to be real-time PCR. Following recent published studies, real-time PCR has proved to be the best method for the detection and quantification of SARS-CoV-2 and water waste. A little is known about the efficacy of SARS-CoV-2 concentration and extractions and identification methods in water waste due to the inhibitors in samples. Next. RNA recovery was conducted using a detection kit for waterway samples. And next. The primes and probes set were described by the US Center of Disease Control, the CDC. These probes target the N1 and N2 regions of the nuclear capsid gene and setting the detection of detecting the envelope E gene as shown in the image below. Waterway samples were multiplexed by 4 target hydrolysis probe-based reverse transcription kits that amplified the SARS-CoV-2 genetic signatures, and the amplification reaction were carried out in 20 microliter reaction volume, and the positive control RNA was run at 1 picogram per microliter. And the SARS-CoV-2 assay reactions included a 10 microliter ITAC universal probe reaction mixer, a 0.5 microliter iScript reverse transcriptase, and a 500 nanometer primus and 125 nanometer probe, and a 5 microliter of extracted RNA template. Next. Using this method, we were able to detect COVID-19 in our waterways around the campus of North Carolina A&T. Samples were classified as positive had SARS-CoV-2 been detected in all 3 sample replicates with a CQ below 35, actually ranging around 35. And next. The number of observed positive waterway samples at each sampling eventually increased throughout the spring semester. Over here, I just highlighted one building that was high in COVID cases that students went and tested. And it shows that individual sampling and testing represented the most accurate measures and active transmission and disease prevalence. Next. This image is just an overview of the sample collection and detection outcomes over the spring. The low flow at collection locations or challenges with positioning of autosamplers intakes were the most common reason for sample collection failure, which is shown in the blue blocks. And these issues were gradually rectified over the course of the semester. And we did have high spikes when it started getting warmer and students were out and about, and data that is not shown here and collected from my health center showed most of that. Next. WBE has emerged as a feasible option for assessing SARS-CoV-2 in various populations in real time. Positive trends allowed us to determine high-risk areas and implement clinical testing to identify symptomatic or asymptomatic patients. The World Health Organization recent studies are indicating a positive correlation between waterways, SARS-CoV-2 RNA concentration, and COVID-19 clinical case reports. Determination of the duration of SARS-CoV-2 RNA in water waste is still not known. This is because water waste tends to get contaminated by industrial waste resulting from disruption of SARS-CoV-2 detections in water waste. And alternative solutions would result in collecting water in the morning peak times, which would enable us to detect the SARS-CoV-2 in RNA water. Next. In conclusion, the correlation between the number of active cases and RNA viral loads can vary due to the delay of viral shedding in stool. These findings do not suggest less efficacy of waterways-based epidemiology. This method is still proving to be efficient and cost-effective in large populations such as college campuses. However, following recent studies, viral load detection in stool has been observed to be lower compared to that of the respiratory tract. And additionally, it has been observed that viral loads in respiratory tracts can be detected onset compared to the fecal matter, which are detected during the peak of the virus. Next. I would like to thank TRISEM for the funding, my PI, Dr. Jeffers Francis, and Dr. Deng, and the laboratory partners at Dr. Jeffers' lab. Next. Thank you. Any questions?
Great talking. It looks like we probably have time for one question. Rachel, you want to go for it?
Hi, Nicole. Great talk. So with the emergence of the Delta plus, and Lambda strains, particularly on college campuses, how would you utilize this WBE system to kind of not only isolate positive versus negative cases, but also which variants might be more prevalent in this building versus another building?
This is what we are trying to implement this semester with genetic sequencing. We haven't gotten into that, but when we do, we will probably be able to mitigate the spread of it quicker and see what variants are evolving around our campus.
Well, thank you.
Awesome. Well, if anybody else has questions, feel free to ask them in the chat. And in the meantime, Rachel, you can go ahead and get your slides going.
Cool, sounds good. I can share my screen.
Yeah, you should be able to.
Okay, can everyone see it?
Looks good, yep. Oh, Rachel, we lost your audio. I'm not sure what happened. Right as you started talking, your audio gave out. Can't hear you right now. No. No, still can't hear. In the bottom left where the audio is, try just switching which microphone and then switching back. Where the mute button is, that little triangle.
Me now?
Yes, now we can.
Can you hear me? Okay, cool.
Yep.
Perfect. Okay, well, try this again. Good afternoon, everybody. My name is Rachel Richards. I am a senior biology student here at North Carolina A&T State University. And this summer, I had the immense pleasure of working in the lab of Sunny Ship, PhD.
Did she freeze? Looks like she froze.
Although tremendous strides has come to the next slide.
Yeah, you're good now. I think you just froze for like 5 seconds, but you're back.
Okay, perfect. Although tremendous strides have been taken to improve water sanitation systems around the world, we're still seeing increased prevalence of clinical burden due to waterborne diseases. Waterborne diseases are actually one of the leading causes of death among children underneath the age of 5 in developing nations. This shows that there is a critical need for more effective treatments and preventative methods for waterborne diseases. Legionella pneumophila is a freshwater bacterium that intrinsically infects amoeba in water, but due to morphological similarities, they can also infect alveolar macrophages in humans. Legionella pneumophila is an etiological agent of the severe acute form of pneumonia known as Legionnaire's disease. The CDC the Centers for Disease Control reports nearly 20 to 40,000 cases of Legionnaires go unreported each year due to the fact that our innate immune defenses can normally clear this infection. However, Legionnaire's disease is still a significant threat to immunocompromised populations. So Legionella, the bacteria, infects alveolar macrophages by way of the type 4 secretion system, which is a molecular syringe that injects nearly 300 effector proteins into the cytosol of hosts. These effector proteins then go and block cytokine production, which disables an effective host action. However, previous studies from the Shin lab have shown that infected cells maintain the ability to secrete IL-1 alpha cytokines, which then go and signal to uninfected bystander cells of the infection and induce them to release other cytokines that are critical to an effective immune defense, namely TNF and interferon gamma. Our preliminary studies from the Shin lab have shown that TNF signaling is enables the macrophages and murine from mice. It enables them to effectively restrict Legionella replication during infection. Indeed, both in vitro and in vivo studies have shown that TNF signaling is critical and even required for effective restriction of Legionella replication. However, we are unsure, we have yet to elucidate the full role of TNF signaling during Legionella infection in human macrophages. Understanding how TNF signaling plays a role in restricting Legionella growth during infection in human macrophages will allow us to understand not only more about how the body clears Legionella infection, but how immune defenses are mounted against intracellular bacterial infections and how they modulate them to be not too overwhelming to damage the body, but to clear the infection effectively. For my project, I proposed a model of TNF signaling that goes and induces activation of a cytosolic PAMP sensor that wants other proteins to then go and mount effective immune responses, namely cytokine secretion and cell death. So based on previous models, I suggest that TNF signaling induces the activation of the inflammasome complex, which then goes and induces further immune defenses to be activated, such as cell death, cytokine secretion, and effective restriction of Legionella. So to understand how TNF signaling restricts Legionella infection in macrophages, I utilized two distinct strains of Legionella 16 to 24 hours post TNF priming. TNF priming, which is exogenous TNF placed in the standard cells. We utilize readouts of cytokine secretion and cell death as induces an effective immune response to Legionella.
Oh, Rachel, you're frozen again. Shortly. She dropped off.
I'm guessing her internet connection is bad and it just booted her.
Is pretty bad. Can you all hear me?
Oh, now you're back. Yeah.
Yeah, I'm sorry about that, guys. My Wi-Fi is pretty bad.
You're good.
Post TNF priming and looked at the growth of Legionella over a 3-day period. As you can see at the 24-hour time point post-infection, there's not a significant difference between Legionella replication, but the 48 and 72 hours post-infection, 2 to 3 days after we had initially infected our macrophages, we see a significant difference in Legionella growth between the macrophages that were primed with TNF versus those that are unprimed in black. What this figure here is showing us is that TNF signaling does enhance the restriction of intracellular Legionella replication in vitro. However, this model cannot tell us the particulars of how the TNF signaling pathway restricts Legionella replication in human macrophages. We utilize ELISA analysis to understand how TNF signaling may enhance or how it induces and enhances cytokine secretion during Legionella infection. Cytokine secretion is key for an effective immune response. So we wanted to see how TNF signaling may promote and increase the amount of cytokine production, namely IL-1 alpha, which we know to be crucial to effective restriction of Legionella. As you can see here at the 4 and 8 hours post-infection or when we infected our macrophages with Legionella, we see that there is significant difference of IL-1 alpha secretion after infection. This suggests to us that TNF signaling does enhance IL-1 alpha secretion during infection to elicit a robust immune response to Legionella. As I had mentioned, cell death is somewhat of a paradigm. Much LDH is being released into the environment as
Rachel, you froze for a bit there.
The cells are infected with Legionella. I froze again.
Yeah, just jump back to, as you mentioned, cell death.
Yeah, so as I had mentioned, cell death is an immune mechanism of infection control. So when cells die, they release lactase dehydrogenase when they die. So we wanted to quantitatively measure how much of this enzyme is being released during the infection, as this would help us to understand how much cell death is occurring in response to Legionella infection and how TNF signaling may enhance how much cell death is occurring due to the infection. So as we can see here, during infection, there's a slightly significant increase in the amount of cell death that's occurring in response to Legionella infection, which suggests that TNF signaling is inducing inflammasome activation, which leads to the cytokine release that we saw before with the IL-1 alpha, and also with the cell death occurring here. However, this is only representative of 2 experiments, and due to some irregularities that we saw, we have to repeat this experiment again to understand how cell death is being utilized and optimized to restrict Legionella replication in human macrophages. Overall, the data suggests to us that TNF signaling may play an essential role in Legionella restriction in human macrophages. We can confirm that TNF signaling enhances IL-1 alpha secretion during Legionella infection. However, further ELISA analysis will help us to understand more about the role of cytokine release during infection and how TNF signaling enhances that. It will be interesting to conduct a Western blot analysis to understand how TNF signaling induces increased IL-1 alpha production versus secretion to see if there is any relationship between the two. Additionally, flow cytometry could be used as a method to understand what other cytokines are being released, are being enhanced and released during infection due to TNF signaling. This will help us understand more about the inflammasome-mediated restriction of Legionella replication on downstream of TNS signaling, and also what may be happening independent of inflammasome activation, but is still dependent on TNS signaling. Additionally, the data has provided knowledge that TNS signaling does induce or does effectively lead to effective restriction of Legionella infection in human macrophages. This also suggests that TNF signaling induces inflammasome activation. However, further studies using ELISA analysis, Western blot analysis, and flow cytometry would help us to elucidate more into the role of inflammasome activation. The LDH assay data that I showed to you suggests that TNS signaling potentially induces cell death in response to Legionella infection as an effective immune defense to Legionella infection. However, continuing using LDH analysis and even PI uptake as a method to see how cell death plays an essential role in restricting Legionella replication downstream of TNS signaling will be helpful. Overall, to recap, for our next steps, we would want to understand if TNS signaling is required for restriction of Legionella infection. This would help us to elucidate more into the role of this particular pathway. Additionally, we want to understand more about the inflammasome downstream of TNS signaling and what inflammasome-dependent pathways mediate TNS signaling during Legionella infection to mediate the restriction of bacterial growth in human macrophages. And finally, we want to understand how TNF signaling promotes cell death as a way to optimally restrict Legionella replication in macrophages. So again, the importance of immune signaling pathways against bacterial infection. While it is key to develop effective therapeutics and treatments to diseases like Legionnaires and the bacterial infections that cause these diseases, understanding more about our innate immune defenses will help us understand how we can provide better treatments to immunocompromised populations. If we understand what naturally occurs in our immune defenses, then we can understand why immunocompromised populations have so much trouble defending themselves against bacterial infections. So investigating the downstream mechanisms of TNF signaling may deepen our understanding of TNF-mediated restriction of Legionella infection in human macrophages. On that tangent, it would help us to understand more about the concerted action of cytokines and other immune defenses that play a role altogether in restricting intracellular bacterial growth and infection. So I want to thank the lab of Sunny Shin at the University of Pennsylvania for funding my research and allowing me to come up for the summer. I want to thank Steve Pollock and Dr. Jasmine Alexander for mentoring me over the summer. And I really want to thank Hong Yu, an undergrad student from the University of Pennsylvania that was instrumental in conducting a lot of my experiments. And I want to thank you all for your time and listening to my non-conventional evolutionary talk, but I still think it's pretty key to understanding how bacteria has changed over the years and how our immune defenses have also changed over decades and millions of years to defend against these pathogens.
Awesome, great talk, Rachel. I think in the interest of time, we'll go right on to the next talk, but I'd encourage people, if you have questions for Rachel, to ask them in the chat, and I'm sure she will be happy to respond there. So for our final talk of the session, Wei is...
Actually, I'm here giving the talk on behalf of who's away on an internship.
Got it. Cool. Go ahead, Kevin.
Great. All right. Thank you so much, Mike. Yeah, thanks for chairing the session. Let me see if I can get my screen share started and working. Share that. I hope y'all can see the shared screen.
Yes, it looks good.
Okay, awesome, fantastic. Yeah, so I'm Kevin Liu, a faculty member of the Department of Computer Science and Engineering. Been really happy to be involved with Beacon over the years. It's just been an amazing opportunity, and it's launched a number of things, including some of the research that I'll be talking about today, which is looking at a new approach to addressing a really fundamental and classical task in phylogenetics and phylogenomics. As an aside, thanks also to the Congress organizers who allowed me to reschedule my talk to today due to a scheduling conflict. So this is a little bit of I guess, a switch from some of the other topics and the other amazing talks in this panel, in the session. Okay, so as I said, Wei is the lead student who's really kind of pushed forward this talk. She's away on an internship, so I'm here to give this talk in her stead. But there's been a number of other students who have really helped push this forward, including my former PhD student Hussein, as well as my current PhD students Julia and Ahmad, as well as a really talented undergraduate student Jack Smith, who's now at Microsoft. And so one of the things that we've had an opportunity to work on, thanks to our support and just involvement in Beacon, is really fundamental and classical tasks in phylogenomics and phylogenetics. One of which is, you can estimate a species tree or phylogeny, and that's a basic thing that's typically done in a phylogenetic or phylogenomic study. But you shouldn't necessarily trust the output of a method. And so really a classical thing to ask is whether or not your estimation is reproducible in the form of trying to calculate phylogenetic support for an estimated phylogeny. And one of the most widely used approaches for addressing this task is a method known as the phylogenetic bootstrap method, proposed originally by Joseph Felsenstein in 1985. And it's at this point really one of the de facto standards for addressing this problem of estimating phylogenetic support. The original paper has accrued something like many citations, and it's now the 41st most cited paper in history. And so the basic problem is this: you're given an input annotation MSA on which you estimate a phylogeny, and what you wish to do is you wish to annotate that phylogeny with support values in the following way. To begin, what the phylogenetic bootstrap method proposes is you simply resample your input multiple sequence alignment columns using the standard bootstrap method, which simply says you take the set of input columns and you sample them uniformly at random with replacement to generate a bootstrap replicate. And then on each bootstrap replicate, you repeat your phylogenetic tree estimation typically. And then what you simply calculate is the proportion for each of the bipartitions in your original annotation phylogeny in the input. You simply calculate the proportion of re-estimated phylogenies across all bootstrap replicates that display that bipartition. That is the phylogenetic bootstrap support estimate for each bipartition or edge in the input phylogeny. And so this is, there's some benefits to this approach, of course, the fact that it's statistically well supported thanks to some theoretical analyses owing to the original bootstrap method proposed by Efron, among other benefits, but also there are some important simplifying assumptions and drawbacks as well. And among these is one that Joseph Felsenstein actually pointed out in his original paper, which I'll just point out here via his quote. He says that a more serious difficulty is the lack of independence of the evolutionary processes in the different characters. For the purposes of this paper, we will ignore these correlations and assume that they cause no problems. In practice, they pose the most serious challenge to the use of bootstrap methods. And so what Felsenstein is talking about here is that when you sample uniformly at random with replacement from the sites in your input MSA, you're essentially assuming that the sites evolved independently and under identical processes. And so this IID assumption is a pretty limiting one because there are a variety of reasons why that assumption really doesn't apply in reality. And these include things like biomolecular structure and function, which can result in sequential dependence within input data, as well as evolutionary processes such as genetic recombination and sequence insertion and deletion processes. And so for all of these reasons and more, this assumption that your data are IID, independent and identically distributed, really is too limiting. And so the use of that assumption, that IID assumption to do bootstrapper sampling to estimate your phylogenetic support, that's ultimately a limitation. And so there have been a number of approaches to try to relax this simplifying assumption. And one in particular I want to point out because we build upon this work. Over a decade ago, Landon and RAWR proposed a pretty nice, simple alternative, which takes the following form. They refer to it as the so-called heads or tails method, And they only looked at this question in the context of MSA estimation, actually, not in terms of phylogenetic tree estimation. And so what they proposed was the following simple trick. Given an input set of unaligned sequences shown here, you simply perform your MSA estimation, you get your MSA estimate, and then their trick was you can read your sequences from left to right and do your estimation, but then hopefully if you read your sequences instead from right to left rather than left to right, you get this mirrored image of your sequences, repeat your MSA estimation on your mirrored sequences, and you would hope that you get the same resulting alignment as output, but you might not. And so to the extent that you don't, this gives you a nice clean reliability check for your original MSA estimation. And so an advantage of this being that, well, you get a nice simple way to check reproducibility for MSA estimation. It also avoids this issue of assuming that the data are distributed IID. You preserve the sequential nature of this data when you're doing this mirroring trick. However, you really only get 2 replicates to analyze. And so to relax that assumption, Landon and RAWR later extended this approach with special purpose parametric methods to deal with this limitation, and only again for MSA estimation. And so really what we wanted to do was go back, take this idea and see if we can propose a more general purpose alternative which kept this nice simplicity, which you could still use not only for MSA estimation, but also for looking at phylogenetic support estimation. And so to proceed, we really made use of several different insights to address this gap. The first one of which takes the following form. Here, I'm just going to show you this first insight using this little example where you've got 8 sequences. They're all of differing lengths. And I actually assume that you have a guide alignment to actually do this resampling. Okay, it's really only for synchronizing for the purposes of the algorithm that we're going to propose, but otherwise it's unneeded for the downstream re-estimation. And so the first insight that we had was, instead of, like in heads or tails, resampling from the start or ends of sequences, I can instead begin this resampling procedure from the middle as shown here with this dashed line. And I can read in either direction. Here in this little example, I'm going to read in the mirrored direction from right to left to the end of the sequences, just like with heads or tails. And so this idea is pretty much the same, except I'm just doing the resampling, initiating the resampling from the interior of the sequence data rather than the ends of it, okay? And so the alignment problem is still meaningful in that case. And so here at the bottom, I'm showing you the resulting set of unaligned sequences for this resampled replicate that you get applying this first insight. The second insight that we had was, hey, if you can read from the ends of the sequence, one end from the sequences to the other or vice versa, then you can simply combine these ideas to just continue this resampling in the other direction if you hit the end of the sequences. And so this starts to look something like a little bit of a random walk, where you've got this reflection at if you hit the ends of the sequences as you're reading. So unlike in heads or tails, when you stop, if you hit the ends of the sequences, here you just keep going in the opposite direction with reflection. And finally, the last insight that we had is that second insight said, okay, if you're willing to reflect at the start or ends of the sequences, why not do so at the interior of the sequences as well? And so you can now have reflection breakpoints inside the sequences in addition. And so what results is essentially a random walk procedure conducted directly on the sequence data. One other technical detail I should point out is that you have to have these reversal breakpoints, and they occur with some probability gamma, which is a parameter of the method. And so we refer to this resulting random walk procedure as so-called RAWR, short for random walk resampling. And so this new resampling method is nice in a few ways. First off, it has this nice kind of simple non-parametric nature, like with some of these other methods, like with heads or tails. It also preserves the sequential nature of data, unlike with Bootstrap resampling. And so to sum up, what we ended up with was this procedure that really synthesizes and generalizes ideas from the Bootstrap as well as the mirrored inputs idea of the heads or tails method. And it's parameterized by a few different things, including the reversal probability gamma, as well as the termination criteria for the resampling itself. And so from there, we set out to study our new method, RAWR, for the application of phylogenetic support estimation using a simulation study as well as an empirical study with empirical benchmarking data. And we found that RAWR resulted in support estimates that offered better type 1 and type 2 error compared to state-of-the-art methods like the Bootstrap, as well as newer methods proposed by others like Gasquel and his collaborators. There is a little bit of a trade-off. So the RAWR approach does result in increased runtime compared to the Bootstrap, but not memory usage. To some extent, this increased runtime trade-off can be offset with reduced replication. That is another thing that we found. And so if you're interested in this, the paper for this did recently appear in the proceedings for ISMB and ECCB just in July. And also those proceedings are published in Bioinformatics. So you can find all this in that publication. To conclude, I really want to just spend the last minute or so of my talk just focusing on the empirical study that was also part of this initial publication. And so our data for this study came from a 2015 study from Lamachani et al., which looked at a really charismatic clade, probably of interest to many, involving Darwin's finches, where this original study was looking at this clade for a lot of different interesting reasons. For us, we simply wanted to basically see if we can look at just part of their original study, looking at just reconstructing the species phylogeny for Darwin's finches and seeing if our support estimation method might give us any clues. And so we reanalyzed their whole genome sequence data from their study, looking at the following species. It's a little bit complicated here, but all I really want to highlight there are a number of species shown here, sampled broadly across the Galapagos Islands and Cocos Islands. Furthermore, the species names are colored according to the different groups under study, which are reproduced here in this estimated tree, where the coloring corresponds here to these different group names, tree finches in green, for example, etc. Here on the left, I've preserved the species names, labeling the leaves of the species tree with the coloring. And the other thing that I want to point out, sorry for the dog barking. I'm just going to keep going. Anyways, so this particular species tree, we actually re-estimated from scratch using our own processing of their original Illumina data. And here also I'm showing Bootstrap support values for our re-estimated tree. And what I can point out is that pretty much everywhere the estimated tree is exactly the same topologically as what's reported in their 2015 study, except for two important bipartitions, which occur here within this tree finches clade. Only within this tree finches clade do we get two bipartitions which differ from what's published in their 2015 study, in the Lamachani et al. Study. And furthermore, the Bootstrap support values are very high within this clade as well, 100%. Everywhere else also, you mostly get high support as well for these concordant bipartitions. That's for the Bootstrap analyses. What do we see for RAWR? The key thing is for those two bipartitions, which differ between our study and the 2015 study, we actually get far lower support using RAWR analysis versus standard Bootstrap support analyses just for this clade. And so what we interpret this as is saying is that there's actually some phylogenetic uncertainty that is occurring here within this clade, and some of that can be attributed due to insertion and deletion processes playing a key role in how these species genomes evolved. And so to some extent, if you're interested in trying to reconstruct phylogenies accurately for this group at least, it's worthwhile to try to factor in alignment uncertainty. In summary, I've just talked about some of our lab's work looking at phylogenetic support estimation using a new approach referred to as RAWR, and we were able to show that RAWR offers some key performance advantages in terms of type 1 and type 2 error compared to the state of the art. In our empirical study, which I highlighted at the very end, we showed that there is, at least in terms of our analyses, some substantial impact of sequence insertion and deletion processes on phylogenetic estimation uncertainty for this very charismatic clade. I'll briefly point out that this is not the only application of random marker sampling that we've explored. There are a number of others as well, including to MSA support estimation, recombination breakpoint detection, and others. I'd like, of course, to acknowledge the support of Beacon, as well as the funding agencies and MSU in support of this work. And thank you for your attention. Thank you for attending this session. And if you are interested in any more information, here's some links to my lab's webpage, as well as a GitLab repository with all of the data and scripts for this particular study. Again, you can find the paper, which was just published in Oxford Bioinformatics, talking about all of this work. Thanks very much.
Great. Well, given that this is the last talk, there's time for questions. So if anyone has questions, feel free to just unmute or put them in the chat. And if not, thanks for coming to the session, and we'll see you later in the conference.
Great session. Thanks, everyone.
Kevin, I was curious actually, I know you mentioned kind of introgression. I was curious kind of what, there are a lot of things that often throw off phylogenetic trees, like introgression, incomplete sorting, gene duplication, these sorts of things. And I was curious which of them you kind of assessed RAWR sensitivity or resilience to in terms of getting more accurate than kind of the standard approaches.
Oh yeah, that's a great question, Mike. Thank you for that. So the short answer in the context of this study was yes, all of those are worth exploring specific to just the empirical study of Darwin's finches. Yes, so introgression is one hypothesis that Lamachani et al. Raised as resulting in uncertainty for their species tree inference. And actually they had conducted a follow-up study where they looked more deeply into that. So we're interested in looking at that just within this tree finch clade of that data set. That's one. Another actually, which is important to point out, is adaptive evolution, right? So this is a very kind of celebrated model looking at rapid radiation and adaptive evolution specific to beaks for Darwin's finches. Yeah, that's another thing which is not accounted for in the context of our analyses, nor of the 2015 study, which we'd like to look at. So that's definitely something for follow-up work. Yeah. Just within this analysis, we're specifically focused on phylogenetic tree estimation uncertainty due to sequence insertion and deletion processes. Yeah, but there's more to look at for sure. Yep.
Yeah. Cool. Thanks.
Sure.
Any final questions? Great. Well, thanks everyone for coming to the session, and we will see you around at later sessions.